Well, good morning, everyone, and thank you for joining us this morning for Winship Grand Rounds. A few housekeeping items. If you're an Emory University or healthcare employee and would like to receive a CME credit hour for attending today, the login information can be found in the chat feature at the bottom of the screen. Also, if you have any issues with the webinar or the CME login, please send Julie Hawkins an email or drop a note via the chat feature. This morning, we're excited to welcome Dr. Georgia Chen. Dr. Chen received her PhD from Louisiana State University in Baton Rouge, Louisiana. After postdoctoral training, she received her first faculty position at the University of Texas Dental Branch as a research assistant professor, and was then moved to a tenure track position at MD Anderson Cancer Center. Dr. Georgia Chen, prior to joining Emory, was associate professor at the University of Pittsburgh, and currently is a professor in the Department of Hematology and Medical Oncology at Emory University. She's a well-known molecular oncologist with a focus on oncogene expression, gene regulation, using animal models for head and neck cancer. Specifically, she focuses on metastases for head and neck cancer models, evaluating anti-cancer therapeutic agents also in this disease. She's known to have established a human xenograft metastasis mouse model for head and neck cancer, which has been used in studying the biology of cancer and in the evaluation of therapeutic agents. So we're very excited to have Dr. Chen this morning, who will talk to us about EGFR targeting. Welcome, Dr. Chen.
Thank you. Yeah, first of all, I would like to thank our leaders to provide this opportunity for me to share my research to the audience. And as a part of my research interest, EGFR targeting therapy is one of the focus in my research since I joined Emory. So I would like to share the data since then. I think everybody knows EGFR is an important oncogene which process carcinogenesis for many solid tumors, including lung, head, neck, esophagus, and colon cancer. So when I joined the Winship, the first thing we did was study the combination therapy with of EGFR inhibitor with other anti-cancer agent. Just give you two examples. First, we studied how the COX-2 pathway interact with EGFR in a preclinical study. We use the COX-2 inhibitor, which is celicoxib and EGFR inhibitor afatinib. Then we found they can interact. They can interact together like a synergistically. Then we moved this preclinical study to clinical trial, phase 1B for prevention of head and neck cancer. And this project was reported in 2014. Yeah, I would say what we have been closely collaborate with physicians, our head and neck cancer oncologists. I highlight their name in those publications. The second example is we use, because celicoxib has toxicity, everybody knows, maybe cause a heart attack, a heart bleeding or something. So we moved to the nature compound. We use a green tea, major components in green tea is EGCG. So we use a combination of EGCG with EGFR inhibitor afatinib. In the same way, we started with a preclinical study. Then this study moved to the clinical trial again, phase 1B, for prevention of the secondary head and neck cancer. So that's part of our publication. So, however, although EGFR has been a target for treatment of head and neck cancer, right now the only inhibitor approved by FDA is Cetuximab. Cetuximab is antibody which blocks the EGFR specifically. But the outcome from this drug, since it's approved in 2016, it shows only only limited responding rate around 13%. And so our research start to focus on to understand the resistant mechanism of the cetuximab in treatment of head and neck cancer. Yeah, everybody also already know, to refresh your memory, EGFR has it's a family. I mean, there is EGFR, HER2, HER3, HER4, they're all family members. The feature of these family members, they can form either home dimer or heterodimer. Then they all have a kinase, most of them have a kinase activity, except the special member, HER3, which is a little bit different from others. Itself, HER3 does not have a kinase activity. It has to be dimerized with others like HER2, EGFR. And also when HER3 dimerized with HER2 or others, their downstream pathway actually is stronger to active PI3-AKT pathway, not the MAPK pathway. But they all share the similar downstream signaling pathways. So based on the literature and our experience, we published a paper to summarize potential mechanism for resistance to cetuximab treatment. Just briefly explain, there are potential, in this figure at least, there are potential 4 types of resistant possibilities. First, if there is any mutation or overexpression of the downstream effectors of EGFR's family signaling, and the cancer cell may bypass EGFR blockade. And second, if there is EGFR dimerization with other receptor tyrosine kinase, such as cetuximab, I'm going to mention that later, and it also share the same downstream pathways. So in this case, if you block EGFR, that may not work. And third type of resistance may come from heterodimerization within the family members. And the fourth possibility, some other pathways, such as STATs pathway and TGF-beta pathway, they all can activate the genes which common to the EGFR signaling pathway. So in that way also, if overactive for those pathways, it may also bypass EGFR blockade. So based on this thinking, we perform our research in the past years. The first thing we studied is the HER3 pathway. I already mentioned HER3 is a little different from other family members. Initially, from TCGA data, we found heregalin, also called the neuregalin, is highly expressed, which has the highest expression level among other solid tumors in TCGA data. So we think maybe HER3 is an important molecule to study. And also, we studied patient samples from Emory. With this time, we focus on the also OPSCC, oropharyngeal patients, we found the overexpression, high expression of the heregulin or neuregulin is contribute to the overall survival. So it's the important thing we need to learn from this pathway. And later on, we found one thing. We obtained two cell lines, one from Dr. Horaro's group. They created cetuximab resistant cell line, and the P is parental, C is resistant cell line. We found, when we studied this cell line, we found they can be, the HER3 expression can be induced after we treat with cetuximab in both cases. So this is a quantified result in the lower panel. So we think maybe we treat cell line with cells with cetuximab, then HER3 pathway may be induced. We also use immunoprecipitation. We found in two cell lines, we found if we treat with cetuximab, we can induce HER2, HER3 dimerization. So that is another evidence to support HER3 participate in the resistance phenotype to the cetuximab. So in this study, we later on, actually, Dongsheng Wang in our lab, he created the shRNA knockdown cell lines called the CH. And then we found that if you knock down HER3, you can also reduce HER2 activation. That makes sense, right? HER2 cannot be activated, and the kinase activity will not work in the HER2. So that also evidence. Okay. Then we treat the three cell lines, parental, resistant cell line, and the HER3 knockdown cell lines with increased concentration of the cetuximab. We found in the HER3 knockdown cell line, originally this SCCHN is resistant to cetuximab, but if you knock down HER3, that becomes sensitive to the cetuximab treatment in different concentrations. So that brings us an idea. If we block cetuximab and the HER3 together in combination, we can further inhibit the tumor cell growth. So the lower panel, panel C, show the evidence of our speculation. The drug at that time we used to inhibit HER3 is called MM121. This is also an antibody, can block HER3 surface, block dimerization of the HER3 to the other EGFR family members. You can see this drug is very potent. In a short time, low concentration, it can inhibit the HER3 phosphorylation, and that is expected that inhibit her p-AKT in a low concentration in 24 hours treatment, and the p-AKT pathway downstream PS6 in two cell lines. We use these two cell lines because SCC47 is HPV positive cell line, and the TU22 is the HPV negative cell line. So we use these two cell lines. We found there's no difference in the inhibition of HER3 in between HPV positive or negative cell line. This is the colony information. Again, we use these three cell lines from colony. You can see the combination show the much better inhibition of a colony formation. If you look at the details quantification, MM121 treatment in the HER3 knockdown cell line, there is no activity, right? Because the HER3 is already out. There is no specific inhibition. But if you use a combination to these 3 cell lines, you can see even the resistant cell line, SCC1C, showed a significant inhibition for colony formation as compared with the control and the cetuximab alone. And when we check the signaling pathway downstream of the EGFR and HER3, we can see the combination can, as we expected, block PAKT in both resistant sensitive cell line and in block the PERK in the combination treatment. But if you see the SCC1C, you can see in the they cannot completely block EGFR at all compared with control. However, downstream, because they do not they may not use EGFR pathway mainly, they probably switch to the in the resistant cell line, switch to the HER3 and HER2 pathway. So in that way, downstream is still being blocked effectively when we use this combination. Based on those in vitro study we're performing, in vivo study, again, we use these 2 cell lines. We use 2 doses of the MM121 high and low, and combination. As expected, the combination showed the best efficacy against the tumor growth. And the lower panel are tumor weight. After we sacrificed the mice, we took the tumor out and then we weighted the same. The result is consistent with the growth curve. And also, surprisingly, not surprisingly, expectedly, SCC1C resistant cell line, when we treat with cetuximab, is not responded at all. But when we do the combination, use 2 dose, high and low, then we see the very good inhibitory effect. The lower panel, we use the HER3 knockout cell line. When we make the xenograft, we treat with cetuximab. You can see without HER2, cetuximab works very well in this SCC1C cell line. So that is another evidence to prove HER3 play a role in the cetuximab resistance. Then we also did a biomarker study. We showed the combination reduced the Ki67 proliferation marker, induced apoptosis, and reduced vasculature formation, which was in CD34 as a marker for the blood vessel. Then further up, we use the patient-derived xenograft to study in this case, we have 6 patients used. You can notice, I want you to notice, I want you to pay attention to the patient 8. We stained the EGFR in those patient samples. We found the patient 8 has lower EGFR expression and different pattern of the staining. Others, they all have like a membrane staining and cytoplasmic staining, which is a very it's classical staining pattern for EGFR. But in the P8 patient, they don't have a membrane staining clearly, and they also have some even nuclear staining. So if you look at the response to the combination, patient 1 is EGFR overexpression cells. Overexpression patient. So they marginally respond to cetuximab, but the combination almost eliminate, wipe out the tumors. In patient 8, you can see these patients did not respond cetuximab. I don't say completely not, but very resistant to cetuximab. However, the combination can sensitize the cetuximab to have some response. So that probably matched the EGFR expression pattern. The others, they all similar to patient 1. And when we check the xenograft tissue, we confirm our observation. When in the single treatment with cetuximab in both cases, we can see the cetuximab induced total HER3 rate. And again, the downstream AKT, as I mentioned, the HER2 HER3, the major HER2 HER3 downstream is AKT pathway, which is over the MAP kinase pathway. So after we publish those data, we move on to the other potential mechanism for the cetuximab resistance. We found that cetuximab treatment not only induced HER3, but also induced c-MET. Based on literature, c-MET can form a heterodimer with EGFR, HER2, HER3. So c-MET may be another mechanism to bypass EGFR blockade. You probably noticed we just published this paper in Oncology recently. Okay, so same idea. We do the combination. This time we tried two c-MET inhibitor, tivantinib and quizartinib, in combination with afatinib. Afatinib is a dual inhibitor which can inhibit both EGFR and HER2. As I mentioned, the HER3 does not have kinase activity. So if you inhibit HER2, you also inhibit HER3. So we think we block, remember that figure I showed in the beginning, earlier in this talk. If you block the surface receptor, which has a potential to heterodimerize with HER3, EGFR, then you probably can block the downstream signaling pathway more effectively. So that shows the combination induce the higher level of the apoptosis in 3 cell lines, including cetuximab resistant cell lines. And when we check the signaling pathway, similarly in HPV positive and negative cell lines, we found the combination, of course, had a better blockage effect on the AKT ERK pathways. But if you look at it carefully, this one, we check the 3 inhibitors for cetuximab, quizartinib, tivantinib, and cabozantinib. And they have a different activity in blocking the AKT and the ERK pathway. So in these 2 cell lines, the quizartinib, cabozantinib showed a better effect than the tivantinib. But in JHU cell line, the tivantinib showed a better blockage than the other. So we provide this information to the clinician for their potential activity of those drugs on the downstream EGFR downstream pathways. So we did the xenograft study, use PDX model again. We checked the quizartinib and tivantinib this time. The purple line, you can see that is combination as compared with a single drug and control that showed better efficacy as expected. Okay, so the third study I would like to mention, remember that figure, the downstream EGFR family downstream may also play a role in bypass cetuximab blockade. So we studied HRAS. HRAS has about 5% mutation rates in the head and neck cancer, so we cannot ignore that. That is one of the important signaling downstream of the EGFR. In this time, we focus on her HRAS because HRAS is also different from other RAS members, different from KRAS, different from NRAS. because the activation of the HRAS require a post-translational modification. An enzyme called farnesyltransferase. This enzyme can make the KRAS being facile translated. So this facile translation is required for activation of HRAS. And there is a drug called tipifarnib. Tipifarnib is a FT inhibitor. So if we block facilation of the HRAS, we will inhibit the function of this protein. Now the literature showed that the HRAS mutated cancer is more sensitive to the tipifarnib than others. But we think maybe white type, this drug also has activity towards white type HRAS. So we performed a study on white type HRAS. And we use, again, the several cell lines. I only show one of them, which is cetuximab resistant cell lines. So we have one control, second tipifarnib alone, third cetuximab alone, and the fourth is a combination. So if you we treat cells in different time points. So if you consider 72 hours result, the combination of tipifarnib and cetuximab can block PEGFR and PAKT and total ERK and the PERK, and then it clearly showed that removed the facilation of the to the HRAS, which is shown in the upper band, which is unmodified. So based on those studies, we performed the PDX study. The first panel A, which is a single drug treatment. These patients do not have a HRAS mutation. However, you still can see the dose-dependent inhibition. Use a single tipifarnib treatment. Then in the next 3 PDX models, unfortunately, we didn't have a cetuximab resistant PDX because when we create the PDX model, that depends on the availability of the surgical specimen. So we did not find the resistant cases to cetuximab. However, we still can see the better. I hope everybody agree with us. We had the p-value calculated. They are significantly better. The combination are still significantly better than the single drug treatment to reduce the growth of the PDX tumors. Okay. Okay. So beyond those studies, I don't know my timing. Beyond those studies, recently we moved to another molecule which called FAT1. And for your information, I would like to introduce what is FAT1, why we are interested in FAT1. FAT1 is a cell surface molecule. It's a cadherin-like molecule which contributes to the adhesion of the cell between the cells and also cell to the stromas. So based on the previous literature, FAT1 may have a tumor suppression function. And although it's not completely concluded, but potentially it is tumor suppressor. In head and neck cancer, FAT1 has the second highest mutation rate as compared with T53. 53 has 72. This one has like 23. This is a TCGA data over like 400 cases. Why we are interested to this enzyme, this protein. I forgot to mention, when we finished the HER3 study, we clearly showed that we block HER3 and cetuximab together, we can inhibit tumor cell growth in the PDX models. And the other people also support our finding. So there's a clinical trial conducted by the company called the CDX. Dr. Nabil Saba is part of this clinical trial. They started the clinical trial use combination of the cetuximab with HER3 antibody-based inhibitor. After phase 1 study, they report in the ASCO, they found all the responder, complete responder, and the partial responder, some of the partial responder, they all have FAT1 mutations. So they suspect FAT1 mutation may play a role in responding to this combination. So Nabil brought this information to us. We start to look at FAT1 in our study. And that is come to the last year about now. The COVID-19 started, closed down the lab. So we start to doing research on the computers. We don't want to wasting time, so we did a study using biostatistician and bioinformatician. We work together on the TCGA data. And this is the TCGA data showed the FAT1 mutation is among the highest, is the highest among other solid tumors in all the TCGA they studied. And also it's marginally, FAT1 mutation is marginally contribute to worse progression-free survival. This p-value is near 0.05, but a little bit higher. So we designed our project based on all the data from TCGA, including clinical data, genomic data, mutation, and the gene expression, mRNA expression, and the proteomics database called the RPPA. There are about 300 protein data collected. Then we performed the bioinformatic and the biostatistical study. This paper has been submitted. Now it's under revision. And we found, it's very interesting, we found the FAT1 mutation has FAT1 mutation is higher in HPV negative. Oh, okay. For this figure, my left panel showed the frequency of the mutation, and right panel showed the expression, mRNA expression level of the mRNA. So if you look at the comparison between HPV negative and positive cases, HPV negative has a higher mutation rate than the HPV positive. And again, the HPV negative cases has a higher expression as compared with HPV positive cases. In overall, all the cases which has the FAT mutation has a lower expression level of FAT than the wild type. The second thing we compare with the location of the tumors. We found the oropharyngeal, oral cavity, larynx, and the hypopharyngeal tumor has a higher FAT1 mutation as compared with oropharyngeal mutation rate. And also wild type has a higher mRNA expression in the wild type than the mutation. But in the oropharyngeal, there is no difference between the wild type or the mutation in terms of mRNA expression. But I would like to mention here, we don't have many HPV positive cases. So I'm not sure this, even it's statistically significant, but we cannot make a completely conclusion here. Further, we found that there's no difference among the stages in terms of frequency of the FAT1 mutation and the expression of the FAT1 mutation, and there's no difference among the node status regarding the frequency of the mutation and the expression level of FAT in all stages of the end status. So this is what we found from TCGA data. And then we compare the protein expression and the mRNA expression between the FAT mutation and the wild type. And then we pick up the significantly differentially expressed protein. And surprisingly, that makes us excitingly, we found the HER3 phosphorylation is induced in the FAT1 mutation and mutated cases, which is highly significant in the total patient and in HPV negative cases. And we found the IRS1, which is insulin receptor substrate 1, which is essential for activation of the insulin receptor pathway, also highly expressed, has a higher expression rate in the FAT1 mutated cases compared with wild type. You can see here, we also compare mRNA. We did not see any result in mRNA. That makes sense because we pick up the post-translational level of HER3 activation, not the mRNA expression. But IRS1 is different. IRS1 already overexpressed in the mRNA level in the FAT1 mutation cases. We also found that cadherin-1 is higher, VEGF2 is marginally higher, and we found that PDL1 is higher in the total in the HPV negative cases marginally, and the HPV positive cases marginally. And we did not see the we only see the mRNA expression in this induced in this protein. However, I mentioned the HPV positive cases are very small. So even the number is good, but we are not completely believed. And also found the CMAT upregulated marginally. And decreasing expression in FAT1 mutation protein, including the ASN1, which is amino acid, one of the amino acid synthetases, and SRC1, PI3K, that makes sense because our mainly FAT1 mutations in the HPV negative cases, right? So this one is not high. And others, I would like to mention PD1. PD1 is marginally lower in the FAT1 mutated cases. And that probably brings us attention for immune regulation of the FAT1. And this protein array do not have, unfortunately, do not have a expression level for the growth factors and cytokines, chemokines, because they're all important for the microenvironment regulation of the tumors. However, we did analysis on the mRNA expression. So not surprisingly, we found the neuregulin, which is the HER3 ligand activator, as highly expressed in the FAT1 mutated cases, which is significant. We only plot the significant cases. And also we found, interestingly, TGF-beta1 is higher in the mutated cases. Then we also found in interleukin-2 is lower in the FAT1 mutated cases. And also we found FGF1, FGF2, they're all lower in the FAT1 mutated cases. And we did a survival analysis, as I mentioned in the beginning. FAT1 mutation alone did not show an impact on overall survival. But if we check the protein which differentially expressed in the FAT1 mutation cases, then we stratified the patient case with FAT mutation and wild type. Then we have 4 groups of the patient. Then we found a significant impact on overall survival, including the HER3, IRS1, and the cyclin E1. They all have showed the impact on the overall survival for the patient. And we look at the HPV negative cases, only RS1 and RET showed the significant impact in overall survival. For progression-free survival, we found the P16 CMAC showed the significant impact on the progression-free survival in the total patient. Then this is a clustering study. We used the protein we identified from the statistical analysis, which significant associated with the FAT1 mutation. We made a heat map. You can see in both mutant and wild type all have clearly 4 subgroups. In one group, group 1, they have a higher EGFR or lower EGFR. They have higher HER2 and lower HER2. And but the HER3 expression, IRS expression is kind of high in this group patient and lower in this one. And it is significant because we already showed in the previous table. Initially, we were a little bit disappointing because white type mutant showed a similar pattern. But if you look at the detail, their pattern is somehow a little different. Yeah, white type has less cases of the EGFR low and HER2 low and HER3 high, right? And those, the lower panel show the difference between, significant difference between these 4 groups. And then we checked the progression-free survival. Then we found a difference. Okay, when I back, the upper band, it shows all the patient cases. So 1 and 3 are from the same patient, 2 and 4 are from the same patient. So we performed disease-free survival analysis among the group 1 and group 2. The patients, as I mentioned, the 1 is the same to the 3, right? So the blue is the subgroup 2, which has lower EGFR and higher HER3, and same to white type cases. If you look at the date of the progression-free survival, the significant difference showed in the 1,200 days, which is about 3 years, in the FAT1 mutated cases. That means if patient has a FAT1 mutation, their progression-free survival appear earlier, then they recurrent earlier. And for the wild type, there is happened, a significant difference happened in the 2,500 days, which is over 5 years, about 7 years. So this patient may have later recurrence. Oh, that may be interesting to your clinician, right? We can probably use the FAT1 as a biomarker. Okay, further study, we compare two more top mutated gene, K-space 8 and P53 in the head and neck cancer TCGA data. We found the FAT1 K-space 8 is co-occur, and 53 and FAT1 are co-occurrence, but the 53 and the K-space 8 mutual exclusive. Further detailed analysis, we will perform further detailed analysis, see how, if the two mutations appear at the same time, what's going to happen. Again, we look at other type of tumor. We found the FAT1 mutation are most likely in the squamous cell carcinoma, including non-squamous and cervical, similar to head and neck. The FAT mutation in lung cancer is about also 23% is similar to head and neck. Interestingly, we found the expression level of the FAT1 at mRNA level and the EGFR mRNA level are positively correlated. That means lower lower expression of FAT has lower expression of the EGFR, and we also know the lower expression of FAT most likely appear in the mutated cases. Okay, this is the important message for us. So to further understanding mechanism, there are many mechanisms behind it, but we start to learn what's happened at the molecular level. So initial thing is we knock down the FAT1 right now in 2 cell lines. We knockdown showed very clearly. We only quantify the band. And we found in both cases, PEGFR was downregulated in the knockdown, FAT1 knockdown cell line. We also found p-ERK pathway is reduced. Signaling in p-ERK pathway is reduced significantly in both cell lines. We found a marginally increase of the p-HER3 and p-HER3 mainly, and the total HER3 we found in one cell line very clearly induced of total HER3. So HER3 pathway may be activated in this case, but we didn't see much induction of p-AKT, so that is surprise. So we'll keep knock out more cell head and neck cancer cell lines to see if we can make a consistent result. But that opened the door for us to perform the further study. Okay, this is the last part, but maybe it's not the least part, it's the more important part of the talk. We want to study the involvement of the cetuximab in regulation of tumor microenvironment. The initial study from our lab, we published in the AACR as an abstract. We found a combination of EGFR, cetuximab, and HER3 inhibitor in combination can reduce PD-L1 expression. So this combination may also reduce PD-L1 to contribute to immune regulation. However, it's a long time, we all know cetuximab has the ADCC effect, which reported by the Bob Ferris for a long time. ADCC effect can activate NK cell. NK cell can secrete interferon gamma. Interferon gamma can induce STAT3. Remember, that is another pathway to bypass EGFR inhibition. So this pathway can induce PD-L1. So if we treat a cell line or tumor with cetuximab, that will be a balance. One try to reduce PD-L1, one try to induce PD-L1 is very complicated. This pathways based on literature already reported. PD-L1 can be regulated by MAPK and AKT pathway. So we did one experiment that clearly showed interferon gamma can induce PD-L1 strongly, and if we use the PI3K inhibitor, it can block this induction. So we think this mechanism is correct, but we did not continue this study because this so many people work on this pathway. Nabel and Christine Chang, they already initiated a clinical trial use combination of cetuximab with the PD-1 inhibitor in combination. So that may already solve the problem of what this pathway recognized. And we start a new study. We think since the microenvironment is important, we started with a screening of the secreted chemokines and cytokines in tumor cells, use a multiplex panel in our core facility. Then we identified one chemokine called the CCL2, also called MCT1, is induced by the cetuximab treatment by combination in another cell line by both EGFR HER3 inhibition and MOL in combination. We confirmed this finding by ELISA. So we think maybe this cytokine, this chemokine play a role in bypass EGFR resistance. Why I will mention later. Also, we found this is not an effect for cetuximab alone. It's effect by blocking EGFR, because even we use afatinib, we can induce expression secretion of this chemokine. So in this study, we use the MOC1 cell line. MOC1 is a mouse oral cancer cell line. Then we use we did the initial like a preliminary study, use the MOC1 Syngeneic animal model. We treat the tumor with afatinib and PF, some big numbers. This is a Pfizer's drug which inhibits the CCR2, which is a receptor of the CCL2. So we block EGFR and CCR2 inhibitor, receptor, we showed better efficacy as compared with single drug. So now comes thinking. We make those picture, we connect those picture together based on the current literature and current funding, preliminary data. We think, right, we may be wrong, right? Everybody can help us to make that figure more logically, right? Based on literature, we know the mutated FAT1 can activate YAP1. YAP1 is a transcription factor, which can induce neuroregulin and HER3. And this is in the some cases are already proved, but not in head and neck cancer yet. And also from literature, people found that when they use transgenic mice knock in, they already express a white type FAT1. They can inhibit a lot of chemokine cytokine, including interleukin-6 and MCP1. That means this FAT1 may modulate the secretion of those chemokine cytokines. On the other hand, neuroregulin or expression will active HER3 pathway. And if we block the EGFR with cetuximab, will also induce HER3 HER2 pathway. And another thing we don't know yet, we found FAT1 activate IGFR signaling pathway. Literature said IGFR can also form a heterodimerization with HER3. So this bypass pathway may further activate the mainly AKT pathway, and maybe MEK, we don't know yet. And this pathway will induce expression, also induce expression of those chemokines, cytokines. So these chemokines, cytokines, they work in the receptor, which is transmembrane, semi-transmembrane protein receptor, and those receptors are also overexpressed in the tumor-associated macrophage and some of the MDSC cells, and these cells contribute to induction of Treg and reduce the activity of the T cells. So theoretically, that is a true case. However, we have to prove it from the experiment. That's a long way to go. However, think about that. If we block immune suppression and the signaling pathways I just mentioned, we may have a better treatment strategy for the patient. So take-home message. We found HER3 as ligand a potentially prognostic marker of head and cancer. If we block cetuximab resistance is related to the activation of the HER2, HER3 cene and other downstream effectors such as RAS. And if we block them together in combination, we may overcome or maybe minimize resistance to cetuximab. And we found later this year, last year, we found the FAT1 may be associated to resistance to EGFR targeting therapy. And finally, most importantly, we have to pay attention to microenvironment when we study the EGFR resistance or resistance to other anti-cancer agents. Okay, finally, I would like to thank you. This is truly a teamwork. Everybody knows now oncology research cannot depend on one lab, one person. It's involved the collaborators. The people I highlighted in red, they contribute the data to what I presented today. And Nabel Saba and Dongshin are my close collaborator. They are head and neck oncologists, everybody knows. And also other people I listed here, I may miss some people's name because of the one slide cannot put everyone on. But they're all very important for our studies in the past. Nationally, I have a collaborator, many from Dr. Chen and Carter Van Weiss in NIH. They all had like expert in the field, and financial support from 3 companies. They provide drug and some small amount of the grant, and also a grant from NCI and Georgia's GCC, and most importantly, WSAT, our institute, continuously strongly support our research effort. I really highly appreciate their support. I stop here. If you have any questions, let me know. Thank you for your patience.
Thank you so much, Dr. Chen, for this excellent talk. I don't think we have too much time. There's 1 minute left to the 8:30 time. But if you have any questions, any pressing questions, you can enter them in the QA feature located at the bottom of the screen. I think I saw one question in the chat box from Dr. Lawson asking, is FAT1 involved in all melanomas or just or mainly mucosal melanomas? Now, I have not looked at, or I personally don't know. Do you know by any chance, Georgia, if the TCGA melanoma data showed any?
I don't remember melanoma is on the list. I don't know. I don't remember because this is mainly involved in the squamous cell carcinomas.
Okay, it would be one question, I guess, to try to explore since this was raised as a possibility. And perhaps Dr. Lossman can communicate offline with us about that.
Yeah, let me check. Yeah, we can check
Sure.
the PCT.
Finally, before we close, I want to let you know that next week Grand Rounds will feature Dr. David Tuveson from Cold Sprung Harbor Laboratory, who will be presenting on the topic of overcoming the 7 deadly hallmarks of pancreatic cancer. You can visit the Winship Grand Round page for these details. I want to thank again Dr. Chen for an excellent talk, and we're going to adjourn given the time. Thank you so much, everyone.
Thank you.